Good morning, students. In our previous lectures, we talked about monoclonal antibodies and the hybridoma technology used for synthesis of monoclonal antibodies. Once the hybridoma cells they have been formed, they are being screened for the desired antigenic specificity, and the screening of the hybridomas will produce antibodies specific for antigen used for immunization. When a hybridoma is grown in tissue culture flasks, the antibody is secreted into the medium at fairly low concentrations, and the monoclonal antibody, it is at much higher concentration it is produced if it is in case of the peritoneal cavity of histocompatible mice. The antibodies, they can be purified from the mouse fluid by chromatography, and the production of monoclonal antibodies involves lot of technical difficulties. And the technical difficulty is that the monoclonal antibodies which are synthesized, most of them which are lacking the human myeloma cells that exhibit immortal growth, they become susceptible to hatch selection but do not secrete antibody and support antibody production in the hybridomas made with them. And as an alternative, the immortality on human B cells, they can be transformed with EPV virus, that is Epstein-Barr virus. When B lymphocytes, they are cultured with antigens in the presence of this virus, some of them acquire immortal growth properties, and these transformed cells, they continue to secrete antibodies. And in this way, the antibody amount secreted is generally very small. So in order to overcome this difficulty, the cells, they are being the monoclonal antibodies, they can be generated in the peritoneal cavity of the histocompatible mice, where excessive amount can be produced, that is 1 to 10 milligram per ml is being produced, and they can be isolated. The density is very high. Another problem which is faced is that antigen activated B cells from the human lymphoid tissues, which are equivalent to mouse spleen, generally human hybridomas, they are prepared from human peripheral blood, which contains only few activated B cells which are engaged in the immune response. So it is possible to obtain B cells that have been activated by antigens present in vaccines that are normally given to people. But human volunteers, they cannot be immunized with the range of antigens that can be given to mice or other animals. To overcome this difficulty, human cells, they are being primed with antigen in vitro conditions. And in vitro, the normal microenvironment of lymphoid tissues is not there. As a result, B cells produces only low affinity immunoglobulin M antibody. So these are some of the problems. So the ways to overcome this is using of the skid mice. Skid mice is the immunocompromised mice. When you talk of priming of human B cells, just to transplant the cells necessary to support human immune response from humans into skid mice. Although normal mice would reject such transplants, but the skid mice does not. And these animals, because they lack the functional immune system. Once the skid mice, they contain both human and B cells, and they can produce the activated human B cells, which can be isolated from the mouse spleen and used to produce human monoclonal antibodies. And once the human monoclonal antibodies, they are produced, they can be used for diagnostic imaging, they can be used for in vivo for detecting or locating tumor antigens, they can be used for early diagnosis of metastatic tumors, they can be used to treat breast cancer cells, they can be used to find out the breast cancer cells when they label with iodine 131 and they are introduced into the blood, and the spread of the tumor to regional lymph nodes can be detected. So this monoclonal imaging technique, wherein the breast cancer metastasis can be studied, this technique used is through immunotoxins. So what are these immunotoxins? The immunotoxins, they are composed of tumor specific monoclonal antibodies which are coupled to lethal toxins, and they can serve as therapeutic reagents. When I say these are tumor specific monoclonal antibodies, they are coupled to lethal toxins, and these toxins they are usually prepared, they are used in preparing immunotoxins like Shigella toxin, diphtheria toxin, all of which inhibit the protein synthesis. These toxins, they are so potent that a single molecule can kill a cell. Each of these toxins, this is immune toxins, it has two types of functionally distinct polypeptide components. So each of these toxins, it is having two functionally distinct polypeptide components. First is the toxin chain, and the second is the one or more binding chains which interact with the receptors which are present on the cell surfaces. Without the binding of the polypeptide chain, the toxins cannot enter into the cells, and therefore it is harmful. So an immunotoxin is prepared by replacing the binding polypeptide with a monoclonal antibody that is specific for a particular tumor. That is specific for a particular tumor cell. So what happens is the monoclonal antibody will deliver the toxin chain specifically to the tumor cells. And when it delivers the toxin chain specifically to the tumor cells, it will cause the death of the cell by inhibiting the protein synthesis. The initial clinical responses to such immunotoxins in patients like leukemia, lymphoma, and other types of cancers have shown promising results, and they are effective too. All protein toxics are working by enzymatically inhibiting protein synthesis. When I say that the protein toxins, they are working by enzymatically, so this shows that catalytical monoclonal antibodies, they act like enzymes. So they are called as enzymes. The binding of an antibody, that is an monoclonal antibody, to its antigen is similar like a binding of an enzyme to a substrate. But in case of binding of this, in cases the binding, it is involving weak, it is non-covalent interaction, and it is exhibiting high specificity and high affinity. But in case of antigen antibody interaction, the antibody does not alter the antigen, as enzyme alters the substrate, but an antibody does not alter the antigen, whereas the enzyme, when it catalyzes a chemical reaction, it alters the substrate. But just like enzymes, the monoclonal antibodies have specificity. They have a specificity, they can stabilize the transition state of a, just like enzyme substrate bond complex, the antibody antigen complex is being stabilized by the specificity of the antibody. It reduces the activation energy just like the enzymes reduce the activation energy for chemical modification of the substrate. There are similarities between antigen antibody interaction and enzyme substrate interaction. For example, a haptin carrier complex. Now what is a haptin? Haptin is an immunogenic compound. When we talk about haptins are immunogenic compounds which are very small in structure. Since they are small in structure, they cannot generate the immunogenic properties until and unless they are bound to a carrier protein. And when they bind to a carrier protein, they have an immunogenic properties, and it is called as haptin carrier conjugate. A conjugate, haptin carrier conjugate, produces antibodies which are specific for haptin, unaltered epitopes on the carrier protein, new epitopes formed by combined parts of both the haptin and the carrier. Haptin cannot function as an immunogenic epitope, but when multiple molecules of single haptin, they are coupled together to a carrier protein which is non-immunogenic or homopolymer, it becomes accessible to the immune system and it functions as an immunodominant determinant. Well, this is something about which I was talking about, a haptin carrier molecule. When I said that there are similarities between antigen antibody interactions and enzyme substrate interactions, some antibodies, they could behave like enzymes and catalyze chemical reactions. Haptin carrier complex, in which haptin structurally resembled the transition state of an ester undergoing hydrolysis. Using this haptin carrier conjugate, the anti-haptin monoclonal antibodies were generated. And once these monoclonal antibodies, they were incubated with an ester substrate, they accelerated the hydrolysis process 1000-fold, and they acted like enzyme that normally catalyzes the substrate hydrolysis. The catalytic activity of these antibodies is highly specific. They hydrolyzed only esters when transition state structure closely resembled that that of the hapten in the immunizing conjugate. The catalytic antibodies, they are called as enzymes, and they have dual role as antibody as well as an enzyme. So with this, I think so I have tried to explain the role of immunotoxin. This is a figure which shows that immunotoxins, which is having two polypeptide chain. One is the polypeptide chain which binds to the surface receptor and makes the toxin enter the cell. Another figure shows the tumor specific antigen which is present, and this tumor specific antigen will bind to the cell and the toxin will enter and the protein synthesis will be inhibited. Immunotoxic mechanisms, it works to kill diseased cells in the body. AIDS therapy as an example, it is using immunotoxin to kill HIV infected cells by raising antibodies that bind to GP120, a viral protein found on the outside of the infected HIV cells. So once the AIDS patients have been treated, the immunotoxin floats around in the bloodstream until it binds to GP120 molecule, which is a viral protein which is found on the surface of the infected HIV cells. Once the GP120 immunotoxin complex gets pulled inside the cell by endocytosis, it is localized, and once it is pulled, it will enter the endoplasmic reticulum, trans-Golgi apparatus in the cell, and to stop the process, kill the cell, disease cell. Now the applications of monoclonal antibodies, they can be used for diagnostic application, therapeutic application, as we talked about immunotoxins, catalytic monoclonal antibodies, enzymes, like we talked about hapten carrier conjugates. Application, diagnosis and therapy. The monoclonal antibodies, they are being used as drug immunoconjugates, they are being used as radioisotopes. In case of breast cancer cells, metastasis is being detected. It is used in measurement of circulation of steroid hormones and in differentiating viral strains. It is identification of tumor cells and they express specific membrane proteins. These are some of the analytical applications of the monoclonal antibodies. And monoclonal antibodies, they are being also used in pregnancy tests. Thank you, students.